Thank you, Yoav, for the optimistic point of view. So we are going to start with the talk of Peter Vogt. Peter, Peter? Not yet. I have to tell a few words about you. So, as you know, we are celebrating the understanding of the molecular genetic basis of cancer. And actually, it all started with transforming viruses, in particular transforming retroviruses. And Peter Vogt was instrumental in increasing our knowledge and understanding of the role of oncogenes, starting from viral oncogene. So this is only partial list of the awards and nominations that Peter was awarded. We can spend practically all the meeting by describing all of them. But I'd like to go back many years to his pioneering and seminal work that actually used electrophoresis of the 60, 70S RNA molecules of transforming and non-transforming retroviruses, the avian leukosis virus and the famous Rous sarcoma virus. And he, together with Peter Duisberg, were able to show that in the slowly transforming viruses or the non-transforming retroviruses, there was a missing piece, missing sequences that actually turn out to be the viral oncogenes. So this is the original experiment, and this is the explanation. When you look at non-transforming retroviruses, such as avian leukosis virus, they have the basic machinery of retroviruses, the GAG, POL, and ENV genes. When you look at rapidly transforming retroviruses, in addition to the basic machinery, they have the additional gene, which is the oncogene, the viral oncogene. In this case, it was SARC. So actually, looking at the amazing career of Peter Vogt, he was involved in major breakthrough. So indeed, he made seminal contribution to the understanding of oncogenes. He was instrumental in the understanding and identification of SARC, the first oncogene, the original oncogene of the famous Rous sarcoma virus that was shown by Rous many years ago to be able to transform animals. He contributed to the discovery of the amazing discovery that actually the origin of viral oncogenes is from cellular genes. And this led to the basic dogma of cancer, where in the majority of tumors, the oncogenes are cellular genes that got some alteration that turned them cancer-causing genes. Peter was also involved in the identification of additional viral oncogenes, such as the famous MYC, JUN, PI3 kinase. All of these were turned later to be very important in pathogenesis of cancer, including human cancer. Peter shown that cancer-causing mutations in the P110, the catalytic subunit of PI3 kinase, is involved in acquisition of oncogenic activity. This PI3 kinase, as you know, is one of the major targets, very prevalent target in cancer. And finally, in recent years, Peter was involved in studies of small molecules that act as inhibitors of the MYC oncogene and thus challenging the dogma that MYC is undruggable. So I'd like to invite Peter Vogt to open this meeting.
Thank you, Gidi, for this very generous introduction. I'm very happy to be here to open this symposium, this Dan DeWitt Symposium on personalized medicine. Personalized medicine is a very flexible concept, and it means different things for different people. Of course, it's personalized cancer medicine, but there is also personalized individual diagnosis. There's a strong personalized element in transplantation medicine. All of these have in common the knowledge of genes. And genetics has had the greatest impact on cancer therapy. And it has now acquired the role of a guiding principle in cancer therapy. What I would like to say here is that the field as a whole is in immense flux. And that was shown very dramatically about a month ago at the annual meeting of the American Society for Cancer Research, American Association for Cancer Research. And they featured a confrontation of two clinicians with very different views. One of them was rather negative, pointing out that only a small minority of patients really benefits from the genetic analysis of their tumors. The other one was forward-looking and said that minority is constantly increasing, and there has been tremendous progress. And we do now have much, much open potential that can be filled, and all the promises that have been made will be fulfilled. Now, cancer is a genetic disease, and that recognition first came with the identification of oncogenes in viruses and became a very general principle when it was found that these oncogenes are really occurring in cells and are derived from cells. And the cellular oncogenes have essential functions, life-preserving functions. These functions can be corrupted by genetic change. And such deleterious changes normally happen just in the tumor, but they can also happen in the germline. And then they are transmitted from generation to generation and are responsible for all the heritable susceptibility to cancer. Now, the field is advancing at an incredible pace. And these advances occur in next-generation sequencing, in proteomics, and bioinformatics. All of them have tremendous importance for personalized medicine. I would like to digress a little bit at this moment and give you two examples, outlier examples, from next-generation sequencing. 3 months ago, the entire genome sequence of the Mexican salamander axolotl was published. And that was a technical feat because the genome of axolotl is 10 times the size of the human genome. And you could say, okay, a technical feat, but does it have any relevance to medicine? Well, it turns out that this animal has the unique ability of regenerating lost limbs. So if a limb is amputated, it will regrow it. And the secret of this may very well be given away by looking at the sequence of the genome. And the other outlier example that I want to mention is in the recent or in the current issue of the Proceedings of the National Academy of Sciences, USA. The entire genome of the tea plant Camellia sinensis. This opens the door to an understanding of the evolution of what is generally considered a very precious cultural heritage of mankind. Now, you could say, well, this is all sort of lateral expansion of genetic knowledge. But the new technology also enables us to go into depth and to interpret sequences in evolution and in function in a way that was never possible before. Also, it is advancing to single molecule sequencing. And single molecule sequencing is now entering cancer care. So the scientists whom we celebrate today have laid the genetic foundation of personalized cancer medicine. Our first speaker, Mary-Claire King, is the pioneer of inherited susceptibility to cancer. Her work has continued to have tremendous impact on the diagnosis, prognosis, and treatment of heritable breast and ovarian cancer. And that work still is the universal guiding post for all investigation of heritable susceptibility to cancer. Our second speaker, Carlo Croce, was the first who identified a known oncogene as a driver of a distinct human cancer. And he established the general principle that chromosomal translocations are indicators and can lead you to oncogenic fusion proteins, discovering many different oncogenes. He's also discovered the importance of microRNAs in cancer. The drug that is currently completely transforming treatment of CLL, chronic lymphocytic leukemia, is based on an oncogene which he discovered. Our third speaker, Bert Ferguson, was the first who took the experimental knowledge on oncogenes and applied it to the analysis of solid tumors, following the development of cancers from an initial lesion to a progressive end-stage cancer. He determined the mutational landscapes of many solid tumors, and that information guides therapy today. He's now involved in harnessing the most recent advances in next generation sequencing and in proteomics in a very, what I would call, virgin field, the field of early diagnosis. And that could have really the greatest impact on cancer survival. So with this, I would like to close, and I would like to ask Mary-Claire to come up and give her talk. Mary-Claire is professor at the University of Washington in Seattle.